Okay, all right. It's my pleasure to introduce Dr. Barry Paw today. I was introduced to Barry Paw by Jerry Kaplan, I think in Prague. I think that was the first time that we met, and I got to hear Barry's stories of using zebrafish to discover genes that are involved in hemoglobinization in the red cell. And you're going to hear some great stories about that today. I just want to tell you a little bit of Barry's history. Barry moved from Thailand?
Burma, yeah.
From Burma
Yeah, close.
to the United States as a small child. His parents were brave and came here with almost no money and established themselves in the United States as immigrants. Yeah. Just making a political statement there. And anyway, Barry went to Berkeley where he, as an undergraduate, he worked in the laboratory of Joe Nealance, who worked on siderophores, iron siderophores, one of the early guys in iron siderophore studies. He then went to UCLA where he got his MD PhD and worked in the laboratory of Liz Neufeld, working on Tay-Sachs disease. And from there, he did a residency at Boston Children's Hospital, and he did a postdoctoral fellowship with Lynn Zahn, where he developed his zebrafish lab, essentially, and has stayed in Boston and has been extremely productive and very collaborative and collegial in the iron and heme field. And that's all I want to say. And Barry's going to talk to us today about mitochondrial and iron and heme.
Oh, okay.
He's also advisor of T32.
He is. Barry is one of the outside advisors of the T32 grant.
Okay. Oops. Oh, disappeared. Oh, okay.
Awesome.
No, awesome. Good.
There's your pointer.
Pointer. Okay. Can people hear me? Or
Let's see it, I think.
I already said there.
Oh,
Okay.
just go straight in.
It doesn't project.
oh, okay. It doesn't project. Okay.
I absolutely will. Let's see. Yeah. Is that okay?
Okay for me.
Awesome.
Let's see. The pointer does it work?
Should. All right.
Oh, there. Okay. Ah. Okay.
Okay.
Okay. Well, it's really a pleasure and an honor to be invited back to present talks, to see friends and colleagues and great collaborators. And it's almost like I feel like this is my second home outside of Boston. So it's a real honor. So today I'm going to be presenting about 3 stories on mitochondrial iron metabolism. And they are, outlined here. So we'll give a little bit of a background on erythroid, why the erythroid system is a uniquely interesting system to study iron and heme metabolism, and that heme synthesis requires multiple steps and transporters. And the 3 vignettes will be a sort of a mitochondrial chaperone, a FAM210B, that's required for iron uptake, sort of a little bit of an old story, a TME14C, which is a transporter of the one of the terminal protoporphyrin intermediates. And a new story that's in the works about, in collaboration with John Phillips here and Hervé Puy on a mitochondrial unfoldase, ClipX, that's involved in regulating heme synthesis. Okay. So my lab is interested in erythropoiesis. One of its hallmark is, as you can see from this cartoon, is from early hematopoietic stem cells, it can differentiate to terminal red cells, undergoing a series of genetic programs that turn on and off genes. But the ultimate production goal is to make hemoglobin, and that is characterized by the globin chains. But more importantly, it's the insertion of the heme prosthetic group that allows you to transport oxygen. So that requires both tetraperol synthesis and iron. So we were interested in what are the iron and heme transporters, and here, this is a review, our understanding of canonical eukaryotic iron transport in invertebrates is that iron basically is bound to transferrin and is bound to its cognate receptor, undergoes endocytosis in the late endosomes, and then undergoes a reduction and acidification that allows you to release iron through DMT1. And then the question is whether, sort of the mystery still is, does it go to the cytosol that Caroline Philpotts has been studying with PCB1 storage in ferritin, and in red cells, does it go directly to the mitochondria? So that's still being studied actively. And several years ago, we showed that mitoferrin is one of the main transporters of iron acquisition in the mitochondria that's in a complex. And once it goes into the mitochondria, it has decision whether does it get stored as mitochondrial ferritin, does it make heme, and then it gets eventually exported to FLVCR, to outside the mitochondria, that then gets complexed to the globins, or make iron sulfur clusters. Okay, the transferrin is then recycled back to the cell surface, and it requires accessory proteins such as sortinexin 3 and Sec15L1, so that helps recycle the transferrin receptor to the surface. So with that, and then the next part of the story is heme synthesis. So as we many people in the audience know, it's a very complicated story where there are many parts, moving parts that involves involvement of the cytosol and the mitochondria. And the first step is the importation of glycine through the glycine transporter. And then, well, I'll talk a little bit about the one regulatory enzyme, ALAS, and the ClipX, which is basically a unfoldase that regulates ALAS stability and activation, which I'll talk a little bit about on the seminar. And the ALS is then exported out through yet unknown transporter. This is sort of still actively investigated. And through a series of modifications, it's then re-imported back into the mitochondria. And then, as I'll talk, one of the transporter is TME14C, which re-imports protoporphyrin 9 back in, and the terminal protoporphyrin 9 is then methylated by iron to form heme. Okay. So the question is, it's a very complicated, but who are all these players that we wanted to investigate? And the approach we did was using genetic screens, many, as Diane mentioned, almost 10 years ago at Prague, or 12 years ago, that we were doing genetic screens in zebrafish to look for many of them that were anemic. And the first one that we worked actively on was a mutant called Frascati, which had hypochromic anemia and erythroid maturation arrest through, in those days, sort of tour de force, I guess it's sort of positional cloning effort that nowadays not people, no anyone in their sane mind would ever do again with exome sequencing. But we classical positional cloning, we identified as the mitoferrin 1 gene was disrupted in these Frascati mutants. And turned out that it was through subsequent functional studies, it turned out to be the main importer of iron for heme and iron sulfur cluster synthesis. It's related to the yeast MRS34 ortholog. So that helped us a lot because Jerry and Roland Lil and for the Belgian investigators, Fauré Foganti, had shown that was probably likely to be an iron importer. So, and in vertebrates, it's required for both primitive and definitive erythropoiesis, and it's such a very conserved function from yeast to higher vertebrates. And then the mitoferrin 2, it seems like there's another paralog that's mostly involved in iron importation in non-erythroid cells, and that Diane and Jerry are working on. Okay. So, and we subsequently showed through proteomics that mitoferrin alone, it doesn't work by itself, that it really is in a complex with ABC10 ferrochelatase that really allows it to efficiently import iron and then to be utilized. So the question now exists is, what are additional players that are involved in heme synthesis? So we collaborated with Harvey Lodish several years ago to look at using bioinformatics. So he had developed a fetal liver culture system that and did deep sequencing RNA-seq to look at candidates. So one of the things at least in mouse fetal liver, you could do, which is erythropoietic, is that you can sort them by CD71 transferrin receptor and TER119, and that allows you to subpopulate a series of erythroid cells based on their maturation from R1, the most primitive progenitors, to terminally differentiated R5. So depending on your different maturation, you can deep sequence RNA, seek to look at what genes are upregulated and downregulated. So as I mentioned, so one of the genes that we were looking for was in this R2 to R3 transition. This was where we found out that based on other known heme and iron transporters, that these most of these were highly expressed. And then we would verify their hematopoietic expression in zebrafish, and then did functional studies using morpholinos to knock down, do they really have a functional consequence on generating anemia, was our initial workout. And then further studies could then be done in mouse embryos and primary cells to really dissect the function in more detail. So that was our strategy. And one of the genes we've been working on is FAM210B, which is very highly upregulated, as you can see, compared to transferrin receptor ferrochelatase, in contrast to housekeeping gene tubulin. So, and what it is a sort of small protein, about 15 kD, has 3 transmembrane domains, and has an N-terminal mitochondrial targeting leader. So likely to be a mitochondrial protein. And so we saw that, having developed the antibody, that it was highly enriched in this R2 to R3 transition, and that it was in the mitochondria by protein expression. Okay? So, and as I mentioned, one of the first things we wanted to do was knock it down in zebrafish, and these are, for those who are not fish aficionados, it's basically antisense oligomers. Instead of having a phosphoester linkage, they have a phosphodiamidate linkage that are resistant to phosphatase degradation. So the rationale was that these tend to stay longer in the cell, and you just inject them in the one cell stage and do either that they target either translation or mRNA processing. The non-toxic is actually an oversell from the company. So this is from the Gene Tools website. And then you can assay for the in vivo phenotype. So, and the first thing we looked at is, as I mentioned, where is FAM210B expressed? And you can see this is a reference GATA1. It's expressed in the ICM, or the blood island, in zebrafish. And FAM210B is also expressed in the ICM, as well as in neural tissues. So when you knock it down at 48 hours, there's a control where you can see benzene positive cells. Knockdown cells are very deficient in the hemoglobinized cells. And this is to be further quantitated a little bit better using a transgenic GFP expressing transgenic animal that expresses GFP under the globin LCR promoter. And you can see there's a control and knockdown of the morphins. You have significantly reduced amount of red cells. So that was a good reassured us that we could go further and investigate. So, and one of the things we wanted to ask is, does it have a iron heme defect? So loading up the cells with iron 55 transferrin, you can see that the amount of iron heme is significantly lower in this is a CRISPR knockout line that we had generated in male cells. And also, when you then measure the amount of iron 55 of just mitochondrial content, it is also down. So indicating that this presumed protein is involved in mitochondrial iron acquisition. So one of the compounds, so at the time we were collaborating with another group at the University of Illinois, Marty Burke. They had done a screen for compounds that are lipophilic, iron chelators, that could bypass the transferrin-mediated iron delivery system. And they initially did a screen in yeast using a FET3 complementation system. And they found that this natural product, that it's been around since the early 90s from Japanese green tea, and it's available from Sigma, can actually accomplish that goal. And you can see it has a sort of catechol type functional group that can sort of chelate iron. And so there's usually about 1 mole of iron to 3 moles of hemeketiol, and that will then basically bypass and correct any protein defects if you have on the membrane cell surface or in the mitochondria. So DMT1, midofferrin can all be basically corrected by this. And it has it has very good specificity in that if you have a defect in heme synthesis, porphyrins, it won't do that. So that was the great thing about this particular compound.
Is it iron specific or does it killing some other metal?
So yeah, very good question. So it does have sort of lower specificity for copper as well, and other transition. But iron is the most.
Copper, I mean, the intercellular was copper 1, that would be copper 2.
2, yeah.
So it would be mainly iron, I would say.
Iron, yeah. It's the physiologic amount was what one of the reviewers asked about that. Would other transition metals be affected? And the argument we made was that what's the more physiologic steady state metal would be iron, and that's why. But other ones, if you really did a KD on it, these other transition metals would probably bind as well. Mm-hmm. So basically, the situation is the model is if you have a transporter that's either on the cell surface membrane or in the mitochondria, you would transport the transition metal across. If you have a defect, iron defect, then the iron basically accumulates, generating a gradient. And then when you give this iron hemeketiol, it basically can bypass this block and therefore complement any deficiency in iron defects. So we tried this in FAM210B compared to wild type, and you can see that control without iron, you have a significant defect in the amount of benzidine positive male cells, and then when you give them hemeketiol, it really complements it back almost to wild type levels. And there's specificity in that, so we think that it's working at this stage. Now, TMEM14C that I'll talk about, that is involved in tetraperol, the transport, we tried that, and so the expectation is you shouldn't be able to rescue. And here you can see that the amount of iron deficiency is low, and when you give a hemeketiol, it really doesn't complement the defect. So indicating the specificity is really good for transition metal transport. So, and this also occurs, that was in cell lines and also in vivo in zebrafish, both the morphins, and when you give it the iron hemeketiol, it really rescues back to almost wild type levels. So and there's also one of the things is that in fetal liver, we also noticed that it's also required for proliferation. If you do shRNA knockdowns of fetal liver for FAM210B, they really don't proliferate, and as well as the amount of enucleation, terminal enucleation doesn't occur as well. So and these are probably side effects of not having being able to make enough heme in these cells. So, and then this is data in collaboration with Jerry and Diane. So we were worried, is this FAM210B a transporter per se in itself? And here is Diane's knockout MEFs for midofferrin 2, and you can see that the survival is much lower because they need more iron for survival compared to wild type. And we were asking, will it complement the defect in these midofferrin 2 knockouts? And it seemed like it does not. So it really cannot function as a substitute, as a primary transporter itself. And the same thing we did with young tau in the MRS34, that it doesn't seem to complement.
So how efficient is this
Ketiol
in suppressing the midofferrin 2 defect?
We haven't looked at in the cells that one, but certainly in midofferrin 1, it's very efficient in terms of like in both in vivo and in our cell line. So basically, the data I don't have it here, but if the male cells that are midofferrin nulls from CRISPRs can be almost rescue to wild type. And then the in vivo with the morphins, you by fact sorting, you can quantify that they go back to wild type levels. So there is you have to be a little bit careful in that in the cell lines there's enough iron in the media that you don't always need to supplement them with iron citrate, but in vivo in the zebrafish because they're E3 media, there's really no source. You have to give it a little bit of iron citrate to just boost the system. So with that in mind, so it turns out that are there where does famotidine interact with other? So we were still mystified, trying to make the story that turns out that we were very lucky by taking a few candidate approach to look at that would famotidine interact with. It turns out that it does interact very specifically with both PPOX and ferrochelatase. So CPOX, another intermembrane protein, really doesn't come down in the pull downs. So seem to be in a complex with these two terminal heme synthesis enzymes. So what here the model we have is that famotidine B exists in a complex that's here. We couldn't demonstrate that it interacts with midofferrin 1 or ABC10, but these are very transient pull downs. But we were quite surprised that it does come down with pretty high degree of specificity. So, and we know that from previous work that midofferrin 1 does interact with FEC and ABC10. So this is sort of our hypothetical model of where this new player sort of fits in this schema. And so it's involved basically involved primarily for iron import to make hemoglobinization, enucleation, and proliferation. So summary one on this one is that it's a novel mitochondrial membrane protein that's upregulated during terminal differentiation, and it's required for as a mechanism to provide sufficient iron for hemoglobin synthesis. So again, what are the other transporters that we were interested in? And we used the same approach of RNA-seq to look for other transporters. And here, using the same strategy, we identified another candidate, TMEM14C. As you can see here, it's highly expressed in the during terminal differentiation, as well with hemoglobin, midofferrin 1, ALAS2, and all the heme synthesis enzymes. So here, TMEM14C turns out to be another very small molecular weight protein. And it turned out there's another related family member, 14A, that seems to have a different expression pattern at the mRNA level. So here, we showed, in fact, we had found this protein through another screen, a complementary screen, besides RNAs. So years ago with Vamsi Muta, to look for another expression screen, this same player showed up in a bioinformatics screen when he went through the database for hundreds of that were available in the public repositories for any genes that were co-associated with heme synthesis. And you can see that TMEM14C is also highly expressed in the blood island, and when you knock it down, you get anemia. So, and so we did a mouse knockout on this one using a gene trap, in fact, ESL system, and here you can see that it's highly expressed in the blood island, as well as in the fetal liver, and in terminally differentiated TUR119 cells where, if you sort them, that TMEM14C protein is highly expressed there. So, and it's a mitochondrial protein in that it's involved in the mitochondrial fraction compared to cytosol. And in fact, when you subfractionate them, it's involved in the inner mitochondrial membrane using mitoplast assays. So what is the role of this? So in this TMEM14C with the knockout, there's very little mRNA. And when you look at the developing a mouse that's E12 and a half, you can see that it's very anemic compared to their wild type siblings. And that when you look at the smears, that they actually have a maturation arrest and they're basically arrested at a very early stage in their development. So, and one of the things we were thinking about was could this be involved in porphyrin synthesis? And one of the things we took chunks of the fetal liver and see if they autofluoresced. As you can see, it is wild type. Here is the TME14C, and you can see the specks of autofluorescent cells, and this is our positive control on the FEC mutant that we were able to get from Luann Peters at the Jackson lab, and you can see similar autofluorescence. So these autofluorescent indicated that that TME14C may be involved in porphyrin biology. And with the help of John Phillips, we were able to measure the amount of where exactly the intermediates that were involved. So here is our wild type, and here's a gene trap knockout. And you can see that this one has coproporogen 3 accumulation, and then on the reverse side is that the gene trap ones have a significant decrease in the amount of porphyrin 9. So, so we were able to at least biochemically place the defect somewhere at this stage in the pathway. So if that will show, if that's truly involved in heme synthesis, we then ask the question, if you were to give it a porphyrin analog due to porphyrin that can bypass this, will it rescue the TME and 14C, the defect? And here is, if you just give iron, it really doesn't rescue the defect. And then with iron due to porphyrin, it really can significantly elevate the amount of heme positive cells in the TME and 14C deficient cells. In contrast, a sortinexin 3 mutant really cannot be rescued, and this was involved primarily in transferrin iron cycling defect mutant. So our model, it turns out that we had proposed at the time that it may be as a transporter that imports protoporphyrin 9 into the mitochondria, and that allows you to then convert eventually to protoporphyrin 9 ferrochelatase and then heme. So are there other potential transporters that are involved? And it turns out I had briefly mentioned that there's TME14A, and this is sort of the NMR solution structure that Clement had published 5 years ago, and you can see they have similar sort of transport helices as transmembrane alpha helices that are colored, and then the non-transmembrane helix gray that probably are involved in heterodimerization. So, so it's possible that in other cells that other family members of TME14 may play a role. Because here in the yeast, one of the we did a bioinformatics search in yeast, and this was the one that was predicted to be a close ortholog of 14C, and I think with the help of Liang Tao, Ali, that we weren't able to show that that they really does not have a significant iron heme defect. So there may be other transporters or genes that may be involved in similar function in yeast.
So I'm wondering, is it possible that it has an importance for so in some cells, CPOX is supposedly in the matrix, and then some on the outside.
On the outside.
So does it matter whether CPOX is matrix versus IMS?
It could be, it depends on that's a good point, yeah. And it may have difference, because you would think that this one, if truly, you should have a defect in the ability to make other targets of like cytochrome assemblies, right? Yeah, and we don't see that as so there may be other redundant genes that may play a role for that can partially compensate for this. Because when we did a CRISPR knockout of TME14C of this guy in male cells, if you have a complete null, they actually are genetically complete null, but they've lost the hemoglobinization phenotype. So we've kept those clones, and our reason, is hand waving argument is could they have suppressor mutations that were selected out? So we will probably want to do sequencing to figure out if there are any overexpression of that 14A or other genes that may be overexpressed to compensate that defect. Okay. So again, summary 2 is that 14C is an inner mitochondrial protein that's essential for erythroid heme metabolism, and in particular heme synthesis defect, and its functions to import protoporphyrin gen 9 into the mitochondria for heme synthesis. So, and then the last story we'll talk a little bit about is genetics of porphyrias, the erythroprotoporphyria. So for majority of the cases of EPP, it's due to a defect in ferrochelatase, which basically catalyzes the terminal metallation of protoporphyrin 9 to make heme. So any genetic defect in that will give rise to EPP. However, there are variants of that that have been described, especially by John Daybach's group, John Charles Daybach, where they showed that there was a C-terminal deletion of ALS2, sort of gain of function. That seemed to account for about 20% of all the, at least in the European and US collection of for EPP causes. And then there's about 5% that's etiology is not known. So in collaboration with Gail Nicolas and Hervé Puy in France, they had identified a proband that had EPP phenotype, but did not have any obvious defects in the candidate genes. So, and it was about the same time that we had published a paper with Tanya Baker on ClipX. So, and it turned out that their candidate gene had a defect, a missense mutation in ClipX. So that's how this story evolved. So this proband, as you can see, the protoporphyrin 9 levels, erythroid protoporphyrin and free protoporphyrin 9 here is very elevated compared to the normal reference values, and that he also had normal ferrochelatase activity. So and so they had excluded other candidate genes like FEC, ALAS2, GATA1, ABC7, mitoferrin. This is the glycine transporter, yeah, and then the ABC10 as potential candidates. And then when they were able to expand the pedigree a little bit, his uncle and father also had elevated erythroid protoporphyrin, but did not have the clinical phenotype. So with that, the whole exome sequencing identified that all of these affected elevated individuals had a glycine to aspartate mutation in this gene called ClipX. So what is ClipX? So it turns out it's basically a mitochondria. So ClipX is a pretty well studied protein that's involved in quality control of prokaryotic and eukaryotic proteins. So there's really two subunits. The ClipX is the unfoldase subunit, and then the ClipP, which is the peptidase that degrades it. So usually there's a tag on these proteins through an ATP-dependent manner. This protein is the target substrate is then unfolded, cleaved through, inserted through the ClipX, and then through further ATP-dependent manner, and this is then degraded. So this has been the sort of the known function of this particular protein for many, many years. Turns out that it has an additional function that one of the substrates, Japanese group showed that was ALS. So and as I mentioned, we got into the story with Tanya Baker at MIT, where it turned out that this mitochondrial ClipX also had an additional function in that it basically is involved in unfolding ALAS so that it allows it to then insert the B6 cofactor into ALAS that allows them to basically catalyze the terminal formation of ALA. And so if you have any defect in this, then you really have a defect in heme synthesis. So we were able to show that you can basically complement the knockdown, the morphins of ClipX by giving them ALA, and that bypasses the anemia. So as I mentioned, the mutation is in a very highly conserved, the Walker A binding motif. As you can see that it's highly conserved from E. coli to humans. And basically, you have lots of activity for ATPase alone. But because ClipX is in a multimer, it's hexamer, if you then add wild type and mutant in a 1-to-1 molar equivalent, it basically acts as a dominant negative to suppress ATPase activity. It doesn't go down to zero, but if you have just all of the units being mutant. So there is some residual ATPase activity in the normal state, at least in the heterozygous state. Okay, so once we knew that, we had tried to overexpress both the wild type and the mutant one in both male cells and adherent HEK293 cells. And with help of John Phillips, we were able to measure the amount of protoporphyrin IX that is accumulated. In wild type, there's a little bit of elevation, but in the mutant one, those that are overexpressing the mutant variant, there's a significantly increased amount of protoporphyrin IX that accumulates. So basically, phenocopying the clinical phenotype. And also, because you have more, that we measured ALAS activity, and at least the mutant one seemed to have more ALAS activity, and that's due to went back and looked at the amount of steady state protein compared to the mutant compared to wild type. There seemed to be much more steady state level of ALAS2 protein. So again, this was a very insightful observation that one of the visiting student from Holland, she kept staring at this her Western blots, and through very persistent. This is sort of an example where you don't ignore what students say to you, because originally we thought, maybe this is just loading control variations, but she was very, very persistent. She brought all her gels and showed sets of blots, and they all had consistent high expression of residual steady state ALAS2 protein.
Very
Yes.
CLPX also interacts with other proteins. Would you expect some other phenotype than protoporphyrin?
You would normally, you would expect, but these patients are fairly mild.
Just pure protoporphyrin.
Well, I haven't examined. We don't have all of the medical records, but its EPP was the only phenotype that we were told that they have. But it is true that there could be other ClipX related proteins that could compensate. Yeah.
And if you just overexpress ALAS, do you see the same effect?
When you overexpress ALAS, do we? So we haven't done that. We haven't done that. So and then when we went back and looked at the original founders, look at their primary CD34 positive cells and look for ALAS2 in the fibroblast, as well as that they also have increased ALAS steady state protein compared to the unaffected brother. And in the CD34 positive cells, you can see that ALAS2 is also highly increased in expression. So then they asked this question, when we looked at, did a pulse chase study to look, is this increased protein due to increased steady half-life using S35 pulse chase approach versus with another complementary is cyclohexamide blockade. By both methods, we could see that the mutant one's ALAS protein has a longer half-life compared to the wild type in the 293 cells, and again in the MEL cells as well. You can see. So by both approaches. So and when you look at the ClipX protein itself, it turns out the mutant one is much more shorter half-life. So it's not able to degrade its substrate, which probably explains why you have more ALAS protein around.
Is that mutation in the Walker B box?
Yes. Oh no, A box.
Walker A.
A.
Oh.
So are there other proteins? Have you looked to see if there are other proteins that accumulate in the mitochondria due to, we'll say, plugging up
the machinery, the quality control? No, we haven't looked, but I'm sure we will hear. If you're asking, then I'm sure the reviewers will be asking similar questions. So I think we will have to get that in our little to-do list. So yeah, that's why it's a great way to vet out this the story now. So here the regulation, basically in the normal state, you have ALAS that's bound to ClipX, ClipP, and there's some sort of equilibrium of degradation, and this enzyme is still active. So you can make ALA, protoporphyrin 9, and heme. So this is our normal scenario. What happens when you have ClipX deficiency? And this was when we had done morpholino knockdowns in zebrafish. You basically have no ClipX, so therefore you really can't degrade. This is all of the ALAS that's there is less active because they don't also have the protoporphyrin, the PLP, the B6 that's inserted. So and you have basically end up with heme deficiency. So and in our scenario, which is the third scenario, you have a dominant negative situation where the mutant ones is highlighted in yellow. Again, it doesn't have ATP activity is reduced, but the enzyme is still active. So therefore, there's more proteins that hang around. This is active, so you end up with more protoporphyrin 9 and porphyria. So that's our simple model. So when we compare the different EPP categories, so this is in our normal state. In heme deficiency, you basically can't form. You have a blockade that's here, so you basically cannot make heme. But there's accumulation of protoporphyrin 9, and the result is anemia with protoporphyrin accumulation. For the ALS2 gain of function mutation, it's a sort of similar scenario where there is more ALAS2 enzyme activity, resulting in more ALA production, more protoporphyrin 9 accumulation, and that could be potentially treated with iron supplementation, as referenced in this paper. And in our scenario, the ClipX mutation has a similar analogy to here, the ALS2 gain of function mutation, and that could be potentially treated with iron supplementation. So we're testing that right now by giving them iron henochidiol to see whether that will reduce the amount of protoporphyrin 9 that comes down. So summary 3 is that ClipX, regulates ALS2 activity by control turnover and its activation. The GE298D mutation disrupts ability of ClipX to degrade ALS, and the resultant stabilization of ALS causes accumulation of protoporphyrin accumulation and a novel form of EPP. So it's probably the first known chaperone mutation to cause EPP. And it's more similar to the X-linked EPP than the more common APP due to deficiency. So, and lastly, I'd like to thank the people who did most of the work in my life: Yvette Yan, Iman Schultz, Kayong Chen, and Lisa Vandervorm, who was a visiting master student from Holland, who really was the one that I give credit for looking at her Western blots to make us go down that path. And a lot of the stuff that we do really couldn't have been possible without having wonderful collaborators. So really, and that's the fun thing of doing collaborations, is really pushing the field. And we have collaborators at Jackson, Luann Peters, INSERM, Gail Nicolai, and Hervé Puy with the ClipX story, at Boston Children's Hospital with Stu, Alan Cantor, Len, University of Georgia with Harry Daly and Amy Medlock, at MIT with Harvey and Tanya Baker, UCLA with Carla Kohler for a lot of the mitochondrial sublocalization, Jim Pallas, which is a wonderful colleague who does a lot of the mouse in situs, and last but not least, our collaborators here at University of Utah, Jerry, Diane, John Phillips, Liang Tao, and Hector, and our funding source from the NIDDK and NHLPI. Right. And thank you for your attention. Yeah.
So, in your system, have you tried any proximity labeling to see whether CPOX is matrix or IMS? Because it seems like that's a real significant unresolved question.
Like
one, does that FAM210, is that bind CPOX, for example?
We haven't tried it, but similar to what Alice Ting had done.
Yeah.
Alice Ting, yeah. We haven't tried that now, but yeah, it's one of the things we could consider doing. Yes, sir.
This is a little bit off topic, but so it looks like from what you were describing that if erythrocyte precursors can't make heme, they don't try to differentiate into erythrocytes. Is that true? And if that's true, is it known how the cell tells it has enough heme and decides whether or not to differentiate?
Yeah, that's a good question. I don't know if people know that what is the sensor that tells it. Do I now apoptose or try eco long as far as I can, but it looks like nucleation is one step that they really can't do. Joe, do you have any thoughts?
Don't know any more than
Yeah. Even though it's a very obvious question, but nobody in hematology knows. Jim, do you know any thoughts?
But there is a crosstalk between heme and globin synthesis, right?
Yes.
And I think Y.W. Khan, when he was blocking globin synthesis, he was stopping differentiation, I thought.
Right, but you need heme in order to get globin synthesis.
No, I understand, that's what I said, but
Yeah.
the question is what stops the differentiation?
There's not much known about the frequency of these mutations in the general population, with the exception of the hypomorph mutation of ferrochelatase. The ALAS gain-of-function mutations have a wide spectrum of disease phenotypes. Some of them get liver failure by the time they're 3 or 4 years old. I wonder if we're going to start finding compound heterozygotes, the gain-of-function deletion mutation of ALAS2, the terminal sequences, coupled with
These things, yeah. I'm sure if you look hard enough, you'll eventually see that.
If you look at the protoporphyrin with the red cells, collection of these patients, from here to here, and the ones that are way out here are the children who get liver failure.
Well, even the uncle and the father seem to have a much milder clinical phenotype, and also the accumulation of protoporphyrin is not as high as the original proband, the index case.
They had much higher FEC activity too.
Compare, okay. Right, well,
Thank you.
Joe. for your attention.
Jim.
Barry.